Okay, welcome to the third lecture for Bio 102, General Principles of General Biology, John Tyler Community College. My name is Mr. Sparks, and I will be your instructor for this lecture. Today we are going to study Chapter 21, The Evolution of Populations. The most common misconception is that organisms evolve through their lifetimes. In fact, organisms do not evolve, it's rather populations. Natural selection acts on individuals, but only if that individual can pass on those traits and those traits are beneficial does that population evolve. Consider, for example, a population of medium ground finches on Daphne Major Island in the Galapagos. During a drought, large-beaked birds were more likely to crack seeds and survive. The finch population then evolved towards the trait of having large beaks by virtue of natural selection. Here is the large-beaked finch of Daphne Major Island in the Galapagos. Here is a graph showing time prior to the drought in 1976. And then the drought was in 77, and then after the drought, we can see that the beak depth or beak length was then increased over time. It was as a result of the drought. So the drought caused a scarcity in food, and the scarcity in food meant that the finches had to survive on larger seeds than they were generally accustomed to eating, and then only the finches that were able to consume those larger seeds did survive, and they passed that trait on to their offspring, and then that affected the population at large. And so the trait of having large beaks was adapted into the population of finches on Daphne Major Island. Microevolution is a change in allele frequencies in a population over generations. Three mechanisms cause allele frequency change: natural selection, genetic drift, and gene flow. Of these, only natural selection causes adaptive evolution. Genetic variation makes evolution possible. Variation in heritable traits is a prerequisite for evolution. Mendel's work on pea plants provided evidence of discrete heritable units, also known as genes. Phenotypic variation often reflects genetic variation. Genetic variation among individuals is caused by differences in genes or DNA sequences. Some phenotypic differences are due to differences in a single gene and be classified as an either-or basis. Either you have this trait or you do not have this trait. Other phenotypic differences are due to the influences of many genes and may vary in gradations along a continuum. Such an example is the coat color of horses. All horses are obviously of the same species. They breed. They have no problem interbreeding and producing viable offspring. But the individual horses can have any number of a different color coats. These different color coats are not, it's not either white or black or brown or Appaloosa or any of the varieties of coats that you could get in the horse. They are varied along a continuum. They vary along a gradation. That's how those traits are transferred. Genetic variation can be measured at the whole gene level as gene variability. Gene variability can be quantified as the average percent of loci that are heterozygous. Genetic variation can be measured at the molecular level of DNA as nucleotide variability. Nucleotide variation results rarely in phenotypic variation. Most differences occur in non-coding regions, the introns, while variations that occur in the coding regions are rarely exchange the amino acid sequence of the encoded protein. So variations Genetic mutations or variations are rare, and where they occur, they often have no effect. So here's a gene. There's the exon portion here off to the left and the intron portion next to it. If those variations, genetic mutations occur in the exon, then it might have an effect on the protein. But generally speaking, the effect of mutations is negligible. Phenotype is the product of inherited genotype and environmental influences. Natural selection can only act on phenotypic variation that has a genetic component. Caterpillars are raised on a diet of oak flowers versus caterpillars raised on oak leaves. You can see here that both of these caterpillars are the same species. They have the same capacity, the same genetic capacity. But that genetic capacity for morphological difference is only awakened depending on the food that they feed on. So the caterpillars that are raised on oak flowers, they tend to look more like oak flowers, which is advantageous because they can be camouflaged in that environment. And same goes for caterpillars that are raised on a diet of oak leaves. These caterpillars, these are cankerworms or inchworms, the same kind that descend from the trees in the springtime. We'll probably see them in the spring semester. They will look more like the oak twigs if they're raised on a diet of oak leaves. So the environment can affect the genetic package that each species contains. Here again is the caterpillar raised on the oak flowers and the caterpillar raised on oak leaves. So the environment affects the potential of the genetic package of the individual species. New genes and alleles can arise by mutation and gene duplication. A mutation is a change in the nucleotide sequence of the DNA. Only mutations in cells that produce gametes can be passed to offspring. These are known as germline mutations. A point mutation is a change in one base in a gene. The effects of point mutations can vary. Mutations in non-coding regions of DNA are often harmless. Mutations to genes can be neutral because of redundancy to the genetic code. The effect of point mutations can vary. Mutations that alter the phenotype are often harmful. Mutations that result in a change in protein production can sometimes be beneficial. Chromosomal mutations that delete, disrupt, or rearrange many loci are typically harmful. Most mutations are detrimental. The duplication of small pieces of DNA increases genome size and is usually less harmful. Duplicated genes can take on new functions and further mutation. For example, an ancestral odor detecting gene has been duplicated many times. Humans have 350 functional copies of the genes, and mice have over 1,000. As you might expect, mice have a more developed sense of smell than human beings do. And that all has to do with the duplication of that odor detecting gene. Mutation rates are low in animals and plants. The average rate is about 1 mutation every 100,000 genes per generation. Mutation rates are often lower in prokaryotes and viruses, but short generation times allow the mutations that do occur to accumulate rapidly in prokaryotes and viruses. In organisms that reproduce sexually, most genetic variation results from the recombination of alleles. Sexual reproduction can shuffle existing alleles into new combinations through three mechanisms: crossing over, independent assortment, and fertilization. You should remember those from Bio 101 when he studied meiosis. The first step in testing whether evolution is occurring is in a population is to clarify what we mean by a population. A population is a localized group of individuals capable of interbreeding and producing fertile offspring. Gene pool consists of alleles that for all loci in a population. An allele for a particular locus is fixed if all individuals in a population are homozygous for the same allele. Here are the two different populations that or two different herds that overlap, forming one population. So here is the porcupine herd to the north, and the 40-mile herd to the left, to the south, and those populations overlap, and those herds overlap and consist of a single population. The frequency of an allele in a population can be calculated. For diploid organisms, the total number of alleles at a locus is the total number of alleles of individuals times 2. The total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual, plus 1 allele for each heterozygous individual. The same logic applies for recessive alleles. By convention, there are 2 alleles at each locus. P and Q are used to represent their frequencies. The frequency of an allele is a population will add up to 1. So P equals the dominant allele and Q equals the recessive allele. And we look at the P and Q together, then necessarily they will add up to 1. Okay, so here is an example of codominant alleles. These are alleles that are incomplete dominance in an allele. So when the red allele and the white allele combine, the heterozygous form is pink. And we use this example of these flowers because it's more easy to imagine the next process that we're going to describe, which is the Hardy-Weinberg equilibrium. So this gives us a visual marker of those organisms that are heterozygous, like the pink at the pink flower at the bottom. So homozygous dominant, homozygous recessive, heterozygous incomplete dominant pink. For example, consider a population of wildflowers that is incompletely dominant for color. There are 320 red flowers, 160 pink flowers, and 20 white flowers. Now we calculate the number of copies of each allele. So the dominant red flowers are 320 times 2 plus 160 pink flowers. So that's 320 individuals plus the 2 or times 2 plus 160 individuals equals 800, and 20 white flowers are okay, so this is incomplete dominance. So these two traits are codominant. I incorrectly stated that one was dominant and one was recessive, but we'll clarify that out in the rest of this exercise. So 20 white flowers times 2, those are each of those alleles there, and then plus 160 is each of these 160 white flowers of these incomplete pink flowers. To calculate the frequency of each allele, P equals the frequency of R equals 800 divided by 800 plus 200 equals 0.08 or 80%. And the frequency of the white allele is 1 minus P equals 0.2 or 20%. The sum of the alleles is always 1. 0.8 plus 0.2 equals 1. The Hardy-Weinberg principle. The Hardy-Weinberg principle describes the population that is not evolving. If a population does not meet the criteria of the Hardy-Weinberg principle, it can be concluded that the population is not evolving. We can also use Hardy-Weinberg principle to determine the number of heterozygous individuals in a completely dominant allele system. The Hardy-Weinberg principle states that frequency of alleles and genotypes in a population remain constant from generation to generation. In a given population where gametes contribute to the next generation randomly, allele frequencies will not change. Mendelian inheritance preserves genetic variation in a population. So Hardy-Weinberg equilibrium describes the constant frequency of alleles in such a gene pool. For example, the population of 500 wildflowers and 1,000 alleles, where P is the frequency of R is 0.8, and P, the frequency of white, is 0.2. So here's an example of a bin with different colored marbles that are going into it. You have a 80% chance of drawing a red marble and a 20% chance of drawing a white marble. Okay, and if you mix these up, the results that you get should describe the Hardy-Weinberg equilibrium. The frequency of genotypes can be calculated. R homozygous dominant equals P squared equals 0.8 to the square equals 0.64. Heterozygous 2PQ equals 2 times 0.8 times 0.2 equals 0.32, and homozygous recessive Q squared equals 0.2 squared equals 0.04. The frequencies of the genotypes can be confirmed using a Punnett square. Okay, so here we have the Punnett square, which shows us the dominant red flower, the heterozygous incomplete dominant pink flower, and the homozygous white flower. So if we cross two white flowers, we get 0.2 times 0.2. That's Q squared. That's 0.04. And if we cross red flowers, we get 0.08 times 0.08. That's P squared, which is what was P squared? P squared, which is 0.64. And if you cross the red with the white flower, you get 0.32, which is the pink flower. So the gametes from this generation, with random mixing these gametes, with random mixing these gametes, you get 64% they should give you 64% dominant red, 32% pink, and 4% white. If P and Q represent the relative frequencies of the only two possible alleles in a population at a particular locus, then P squared plus 2PQ plus Q squared equals 1. Where P squared and Q squared represent the frequencies of the homozygous genotypes, and 2PQ represents the frequency of the heterozygous genotype. P squared is the frequency of the dominant genotype. 2P squared is the frequency of the heterozygous, and Q squared is the expected frequency of the recessive genotype. That should add them all up equal 1. The Hardy-Weinberg theorem describes a hypothetical population that is not evolving. In real populations, allele and genotype frequencies do change over time. The 5 conditions for non-evolving populations are rarely met in nature. They have to be no mutations, random mating, no natural selection, extremely large population size, and no gene flow. It rarely occurs, but it does. We'll see an example here soon where we can see where it does occur. Natural populations can evolve at some loci while being in Hardy-Weinberg equilibrium at other loci. So Hardy-Weinberg equilibrium can be effective, it can be in effect for certain traits and not in effect for other traits. Some populations evolve slowly enough that evolution cannot be detected. We can assume that the locus that causes phenylketonuria is Hardy-Weinberg equilibrium. Given that PKU gene mutation rate is low, the mate selection is random with respect to whether or not the individual is a carrier of the PKU allele. A PKU it's like an allergy. It's a reaction to part of the chemical component of aspartame, and you'll see the warning on the side of soda cans and other foods that contain certain artificial sweeteners that they say phenylketonuria, beware. This is the population that we're talking about. So PKU gene mutation is low, and mate selection is random with respect to whether or not an individual is a carrier of the PKU allele. Natural selection can only act on rare homozygous individuals who do not follow dietary restrictions. So in other words, if you consume that aspartame and it winds up causing you a terrible reaction, then it would affect you with natural selection. But if you follow dietary restrictions, then it's not going to have any effect. Okay, and the population is large. We're talking about the human population at large, which is pretty large, about 7 billion at this point. Migration has no effect, whether as many other populations have similar allele frequencies. So just because you're going to travel, the populations travel from one area to another into mix with another population, the rate of PKU is relatively the same in any human population wherever you travel. The occurrence of PKU is 1 per 10,000 births. So that's Q squared equals 0.0001, or Q equals 0.1. The frequency of normal alleles is P equals 1 minus Q. 1 minus 0.01 equals 0.99. The frequency of carriers is 2PQ. That's 2 times 0.99 times 0.01, which is 0.0198. So that's the carriers, or the heterozygous population, or approximately 2% of the U.S. population. Natural selection, genetic drift, and gene flow can alter gene frequencies in a population. 3 major factors alter allele frequencies and bring about most evolutionary change: natural selection, genetic drift, and gene flow. Natural selection. Differential successes in reproduction result in certain alleles being passed to the next generation in greater proportions. For example, an allele that confers resistance to DDT increased in frequency after DDT was used widely in agriculture. Genetic drift. The smaller a sample, the more likely it is that the chance alone will cause deviation from predicted result. Genetic drift describes how allele frequencies fluctuate unpredictably from one generation to the next. Genetic drift tends to reduce genetic variation through loss of alleles, especially in populations. So what we're talking about here is like migration events where a small number of individuals will travel to an island, for example, and then only those organisms that migrated will carry the traits, and they have a limited set of traits. It's less than the larger set of traits of the original population that they came through. And then also there's another effect related to genetic drift called bottlenecking. And genetic bottlenecking is when you have a large population and many of those organisms get killed, or the population is reduced in size, and then only the survivors have those genetic traits. And that is those are 2 major functions behind genetic drift. So population migration and bottlenecking. I believe we'll talk about them a little bit later on this lecture. Okay, so here's an example of a population. The ones that are highlighted in white are the ones that survive and generate offspring. Those 5 plants leave offspring, and of those 5 plants, only 2 are surviving, and then of those 2 that survive, the traits are only the dominant red trait. So basically, you have a mixed population here, and then because only a few survive and they live to produce offspring with limited traits, and then only those two organisms with those limited traits are surviving to produce offspring, and then they produce a homogeneous population that carries only those traits. So that is how genetic drift functions. Excuse me, I had to deal with my cell phone here. Okay, the founder effect. The founder effect occurs when few individuals become isolated from a larger population. This is like the migration scenario that I was telling you about. The allele frequencies in the small founder population can be different from those in the larger population due to chance, just in the example that we were talking about before. The bottleneck effect can result from a drastic reduction in population size due to sudden environmental change. By chance, the resulting gene pool may no longer be reflective of the original population's gene pool. If the population remains small, it may be further affected by genetic drift. Okay, so here's the genetic bottlenecking event. It happens with big cats because they have relatively small populations spread over a large area. The original population may carry a mixture of different traits. Then there's the bottlenecking effect, like the Florida panther was killed off over time by ranchers and people that were that were afraid of the animal. Those ones that survived only had limited traits. You can see in the final container here. So here's the original population, many different traits, the bottlenecking event, the destruction of the populations, and the surviving population then can only reproduce with those limited traits. Okay, so by chance, blue marbles are overrepresented in the surviving population. Bottlenecking event, surviving population. Okay, what's interesting to note is that the Florida panther is it has many inbred characteristics, characteristics that are that were noted by Fiona Sundquist, a biologist who studies large cats, and they found traits like that were believed to be distinctive of the Florida panther, the short face, and there's a kink in the tail, and other minor phenotypic trait distinctions. And those also happen to be traits that occur when you inbreed domestic cats. So people that are breeding Persians or different breeds of domestic cats, those cats can have some of the same traits. They're bottlenecked by virtue of artificial selection, and the natural and the puma, the Florida panther, is bottlenecked by virtue of hunting pressure and its isolation in the Florida habitat. Understanding the bottleneck effect can increase understanding of how human activity affects other species. Just for example, I was talking about the puma, the Florida panther, and how it was affected by human habitat fragmentation and hunting pressures. Habitat fragmentation is the developing of land into farmland or into cities and towns and what have you, areas that are no longer suitable for the habitation by the Florida panther. And those reduction of habitat can affect the genetic diversity of those species. The impact of genetic drift on the greater prairie chicken. This is a classic example. The loss of prairie habitat has caused a severe reduction of the greater prairie chickens in Illinois. The surviving birds had low levels of genetic variation, and only 50% of their eggs hatched. So reduced genetic variation does have an impact on particularly on species that are rare or endangered. Okay, so here's Illinois. It shows the previous range of the greater prairie chicken, and now it shows the current range of the greater prairie chicken. So you can see here's a large population, covered a large area, and now it's only 2 isolated populations remaining. Okay, so here in the location we showed the estimated population size, versus the here's in Illinois, and then in Kansas and in Nebraska. These are populations that had no bottleneck. They have the number of alleles per locus is reduced in the 1993. The present day population, you can see it went from 5.2 to 3.7. It still remains high in Kansas and Nebraska, where there were no genetic bottlenecks. And the percentage of eggs hatched, they were 93% and 99% and 96% in before the genetic bottlenecking event. And then after the genetic bottlenecking event, the reduced genetic variability, they have the 50% or less percentage of viability. Okay, so here's a better image. It shows the range of the greater prairie chicken in Illinois, and then there it is today after 1993, much smaller population. Okay, here's the table in greater detail. Just what you want to take from this is the pre-bottleneck percentage of eggs hatching, 93%. The post-bottleneck percentage of eggs hatching is 50%. And here's the greater prairie chicken. It's quite a bird. They like to do this little dance. The males do a little dance around the area where they live, where they try to attract the female. That is the area where they try to attract the female is known as a lek, and the behavior is known as lekking behavior. And they do a little dance and they try to attract the female. And if they're successful, they fertilize the female, and the female will go off and have her eggs. Researchers use DNA from museum specimens to compare genetic variation in the population before and after the genetic bottleneck. The results showed a loss of alleles at several loci. Researchers introduced greater prairie chickens from populations in other states and were successful in introducing new alleles to increasing the egg hatch rate to 90%. So by understanding genetic variability and genetic drift and bottleneck effect, we're able to undo those effects and increase genetic variability and improve the survivability of the species. Genetic drift is a significant in small populations. Genetic drift can cause allele frequencies to change at random. Genetic drift can lead to a loss of genetic variation within populations. Genetic drift can cause harmful alleles to become fixed. Gene flow consists of the movement of alleles among populations. So gene flow is kind of the opposite of genetic drift. Alleles can be transferred through the movement of fertile individuals or gametes, and gene flow tends to reduce genetic variation among populations over time. Gene flow can increase the fitness of a population. Consider, for example, the great tit, Pars major, on the Dutch island of Vlieland. Immigration of birds from the mainland introduces alleles that decrease fitness in island populations. Natural selection reduces the frequency of these alleles in the eastern population where immigration from mainland is low. The central population, high immigration from the mainland overwhelms the effects of selection. Okay, so here's the gene flow. Females born in the central population versus the eastern population. It seems that so females born have a greater chance of survivability in the central population, whereas the survival rate is still high in the eastern population, but the population is less adapted to the eastern population. So here what we see is the the eastern population here is getting influence from the mainland, more influence from the mainland than the central population, and it's getting more influence from the mainland, and it's getting these birds were moved from one population to another, and the population shows that those born in the eastern section were more successful. They had a higher survival rate in the eastern section, and those born in the central section had a lower survival rate. Gene flow can increase the fitness of a population. Consider, for example, the spread of alleles for resistance to insecticide. Insecticides have been used to target mosquitoes that carry West Nile virus and other diseases. Alleles have evolved some populations that confer insect resistance to these mosquitoes. The flow of insecticide-resistant alleles into a population can cause increase in fitness. Gene flow is an important agent of evolutionary change in modern human populations. Natural selection is the only mechanism that consistently causes adaptive evolution. Evolution by natural selection involves both chance and sorting. New genetic variations arise by chance. Beneficial alleles are sorted and favored by natural selection. Only natural selection consistently results in adaptive evolution, an increase in the frequency of alleles that improve fitness. Natural selection brings about the adaptive evolution by acting on an organism's phenotype. The phrases struggle for existence and survival of the fittest are misleading as they imply direct competition among individuals. Mostly what natural selection relates to is how the individual fits into the environment. Reproductive success is generally more subtle and depends on many different factors. Relative fitness is the contribution of an individual makes to the gene pool of the next generation, relative to the contributions of other individuals. So, for example, if all your friends are having 2 kids and you have 10 kids, then your relative fitness is greater. Okay, so that's relative fitness is basically the amount of offspring that you contribute to the gene pool. Selection indirectly favors certain genotypes by acting directly on phenotypes. There are 3 modes of natural selection. Directional selection, which favors individuals at one end of the phenotypic range, and disruptive selection, which favors individuals at both extremes of the phenotypic range, and stabilizing selection, which favors intermediate invariants and acts against extreme phenotypes. Okay, so here is your population, and we'll say that it has a general phenotype which favors the intermediate phase between light-colored and dark-colored. This is going to call our original population. And then directional selection is where, let's say that these mice, they live on a dark-colored habitat. And because of that dark-colored habitat, birds of prey are able to find and eat the light-colored mice. So they act as a directional selection. The predatory selection pushes that evolved population into the darker-colored phase, where the darker-colored mice are more successful on the darker-colored substrate. Okay, disruptive selection is where these phenotypes, the selective pressure may come at an intermediate stage, and for some reason the mid-colored, the mid-range tan-colored mice are are more evident to predators, and the light-colored mice are able to hide in the light-colored parts of the habitat, and the dark-colored mice are able to cover in the dark-colored parts of the habitat. And the disruptive selection acts against those mid-color, the tawny mice, and it pushes the selection to favor either of the two extreme phenotypes. Now, the opposite of that can also occur, and that's called stabilizing selection. And that's where the environmental factors are favor against the dark-colored and the white-colored mice, and they are the mid-colored, the tawny-colored mouse is the one that is then favored by virtue of surviving the natural selection. Okay, a key role of natural selection in adaptive evolution. Striking adaptations have arisen by natural selection. For example, certain octopuses can change color and even shape readily for camouflage. For example, the jaws of snakes allow them to swallow prey that is larger than their heads. So snakes have adapted to be able to dislocate their jaws, and they can open their mouths so they can consume animals that are larger than themselves. Okay, so this is like a small river snake. Can consume a fish that is much larger than itself because it's able to disconnect its jaws and open them up to consume that organism. Okay, here we even have an illustration. All right. Oh, whoops. Here's the ligament of the jaw that's evolved in order to help to open that jaw. It's pretty much disconnected from the rest of the jaw, and it's able to eat an egg. Okay, so this snake here can open up its mouth, and that's an adaptation caused by natural selection that enables that snake to increase its survivability. Natural selection increases the frequencies of alleles that enhance survival and reproduction. Adaptive evolution occurs as the match between an organism and its environment increases. Because the environment can change, adaptive evolution is a continuous, dynamic process. Genetic drift and gene flow do not consistently lead to adaptive evolution, as they can increase or decrease the match between an organism and its environment. So genetic drift and gene flow, they occur in populations, but they don't generally lead to adaptive evolution. They can decrease the effectiveness of a population, and I guess that might increase adaptive evolution with those populations then no longer survive. Sexual selection. Sexual selection is the natural selection for mating success. It can result in sexual dimorphism, marked differences between the sexes, and secondary sexual characteristics. Like secondary sex characteristics between a hen and a rooster. A rooster has a much larger comb, and it's got longer, more showy tails. Or a peacock is a great example. A peahen versus a peacock. Peahen is kind of drab, rather gray colored, and the peacock you may be familiar with is purple and blue and green, and it's got quite a long tail that's amazing in its splendor. Okay, so here we have a peacock. Here's a peacock and a peahen, and you can just take a look at the difference between the peacock and the peahen. The job of the peacock is to try to attract the peahen as a mate. That's why it's got all those big long feathers, and those feathers are even evolutionary disadvantage because it makes it easier for the predators to catch the peacock. But you can tell that it's worth it to the peacock. The species has evolved and it's able to contend with that detriment, and it's necessary for the male to woo the attentions of the female peahen. Intersexual selection is competition among individuals of one sex, often males, for mates of the opposite sex. Intersexual selection, often called mate choice, occurs when individuals of one sex, usually females, are choosy in selecting their mates. Male showiness during to mate choice can increase a male's chances of attracting a female, while increasing his chances of survival. How do female preferences evolve? The good genes hypothesis suggests that if a trait is related to male genetic quality or health, both the male trait and the female preference for that trait should increase in frequency. Okay, so here are some gray tree frogs. The gray tree frog have different traits. The one has a certain type of call, and the other has another type of call. And the female gray tree frog will select for one or the other of the species. So let's see if I can find out where this is. Okay, so the offspring of the LC gray frog was able to had a greater survival rate than that of the SC green frog. So in theory, the female gray tree frog should select the LC male's call, the LC by rate of its call. Okay, so in 1995, the LC had better survivability. In 96, had better larval growth, better shorter time to metamorphosis. So all these characteristics suggest that the offspring of the LC male was improved, so that the female should be selecting for the trait of the call of the LC male. So the LC male's got the good call, it's got the good genes, it has the better offspring. The female wants that, so selects for that trait, secondary sex characteristic of the male. Preservation of genetic variation. Neutral variation is genetic variation that does not confer a selective advantage or disadvantage. Various mechanisms help to preserve genetic variation in a population. Diploidy maintains the genetic variation in the form of hidden recessive alleles. Heterozygotes can carry recessive alleles that are hidden from the effects of selection. They only become visible or apparent under certain conditions of natural selection that would select for those hidden recessive traits. Balancing selection occurs when natural selection maintains stable frequencies of two or more phenotypic forms in a population. Balancing selection includes heterozygote advantage and frequency-dependent selection. Heterozygote advantage occurs when heterozygotes have higher fitness than do both homozygotes. Natural selection will tend to maintain two or more alleles of that locus. For example, the sickle cell allele causes deleterious mutations in hemoglobin, but also confers malaria resistance. Okay, so that's an example of heterozygote advantage. So the sickle cell trait in a heterozygous advantage, in a heterozygous individual, you've got the sickle cell trait and the normal cell trait. But if you just have the recessive sickle cell trait, then that causes sickle cell anemia, which decreases the ability of the red blood cell to transfer oxygen. Okay, here is all frequencies of sickle cell anemia, of sickle cell trait. It occurs in areas, you can see here are areas affected by the Plasmodium falciparum, the malaria protist. Okay, occurs all over this region, but the sickle cell trait is most frequent in West Africa and in parts of India. And this trait helps people to adapt to that sickle cell. If you have the heterozygous advantage, you're adapted to survive that malaria. If you have homozygous dominant, then you don't have the sickle cell trait, and you're not adapted to resist malaria. And if you're homozygous recessive, then you have both sickle cell traits, and your ability to transfer oxygen is compromised. Frequency-dependent selection occurs when the fitness of a phenotype declines if it becomes too common in the population. Selection can favor whether phenotype is less common in a population. For example, frequency-dependent selection selects for approximately equal numbers of right-mouth or large-mouth scale-eating fishes. So depending on the environmental conditions, different phenotypes can be promoted. So what these scale-eating fishes are, they swim up alongside of another fish in these they're cichlids, and they do this in this lake in Africa. They swim up alongside the fish, and then they eat the scales off the fish. Well, the fish eventually becomes aware of that, and it will start to defend the left side where it's always getting sneaked up on. And then the left side scale-eating fish will be they won't have an easy time of sneaking up. So then the right side, the right mouth scale-eating fish will be more successful. And then the population will become, the prey population will become more aware of that, and then it'll select against that the right side feeding fish, the scale-eating fish. Okay, so here we have, here's a population of right mouth and left mouth scale-eating fishes, and they swim up on these fish here. Sometimes the populations are skewed towards the left-eating fish, and sometimes they're skewed towards the right-eating fish. So the left-eating fish, over time, it has a decline, and then as time passes, its shape becomes more selected, and it increases until it reaches a height where the prey species are adapted to resist its attacks, and then it declines again, and then it increases. So it declines and increases, declines and increases over time, depending on depending on the prey fish and how it's reacting to the attack. Why natural selection cannot fashion perfect organisms? Selection can act only on existing variations. Evolution is limited by historical constraints. Adaptations are often compromises. Chance, natural selection, and environment interact. Okay, here's a bat eating a frog. Let me go back to that. I just want to stress these again. Okay, so natural selection cannot fashion perfect organisms because it's only operating within the constraints of the phenotypes of those given organisms. Selection can only act as on existing variations. Natural selection doesn't create new variations, only mutations can do that, but then those mutations are in the population, they exist as existing variations. Evolution is limited by historical constraints, so it can only act on existing variations. Evolution can only act on something that has been in the population. Adaptations are often compromises. So an adaptation may be good in one respect and bad in another respect. People, let's see if I can have a good example here. The back of the human being enables it to stand upright and to see out over farther distances than it would if we were stuck on all fours. But at the same time, there are structural differences of the back. If anybody has back problems, you know that you can sympathize with this. But because we've gone upright, we've lost certain strengths in our back. So we've gained some ground by being able to see over the horizon, and we've lost some ground by the weaknesses in our backs. Chance, natural selection, and environment interact. So the chances of different mutations, natural selection, the environmental impact, they all interact to produce organisms that live in that evolve into the habitats that they exist in today. Here's a bat eating a frog. I'm not sure why this is here, but okay, so it says the loud call that enables the Tungara frog to attract mates also attracts some more dangerous characters in the neighborhood. In this case, a bat about to seize its meal. So the bat has an adaptation to call its mate, but it is dangerous because it also calls in the predators. I'm sorry, the frog calls its mate, and it calls its predators. Okay, so this is the Hardy-Weinberg equation used to interpret data and make predictions. This is on page 406. It's more lengthy than I can go into. Here are the different populations: directional selection, disruptive selection, stabilizing selection, all different types of selection involved in natural selection. Okay, here is some mussels. These are the genetics of mussels, freshwater mussels that were adapted to different salinity rates in Long Island. And so what this shows is that we're able to identify a particular allele, the LAP94 allele, and this is an allele that is used to metabolize salinity. And as you can see, as the environments get more saline from one inside the Long Island Sound, where it's brackish water, to ocean salinities number 11, as we get increasing salinity, that LAP4 allele is more prominent in the population.